everyone. In here, my name is Marchellina with the student number is 082114153011. In here, I want to say about my thesis proposal with the title is The Abundance and Characteristic of Microplastic from East Java Coastal and Scatopagus argus orcans. Here's our first topic. As a human, we always produce a waste product. Every day, we use a plastic in our life. And this is an information that 10% of our waste product is a plastic. Based on the LIPI in 2020, in Indonesia, there is increasing number of using plastic for daily life, especially in package and wrapping food. And the plastic waste can be end up in the coastal. As we know, Indonesia has a big coastal, and then two of it is the north and south coastal in Java, Indonesia. In the coastal, there is some organism that live there and made the coastal as their natural habitat. One of it is the fish, with the name is Catophagus arcus. Some the researchers said that fish is one of the organisms that contaminated by plastic debris. And then the second topic is about what is microplastic. So microplastic can be made from degradation of macroplastic that we always use in our daily life. Or yeah, some factory already made by the tiny size. The size is less than 5 millimeters, but in my research in here, we use 5 millimeters until 1 millimeters. The microplastic have a specific colors that they are different with the organ and then specific shapes like a fiber or a fragment or a pellet, and then the specific polymers that we can use FTIR to detect it. And the bad news, the microplastic can be a contaminant in our environment. It can be accumulated in some organism, and one of the organism is fish, and then we always eat fish for our daily life, so it can be go to the biomagnification. And then in here, the third is the reason why we should do this research. The first, we want to know about the microplastic on our commercial fish in Indonesia. For the example is Catophagus argus. And then the second is the correlation between the microplastic abundance in the water and the organ. For example, we will collect for the fourth place, they are Lumajang, and then Sidoarjo, Gresik, and Probolinggo. And for the organ, we will use respiratory organ and digestive. The third is the new information about the microplastic in commercial fish in East Java coastal Indonesia. And the last, we wanted this information being the reference to make a new regulation about the microplastic in our environment. To do this research, we need some material and tools. For the examples like a water sample and the fish, Scatopagus arcus, hydrogen peroxide, plankton net, oven, compound, Watman, microscope, and FTL. And the next is the step to do this research. The first is we're collecting the sample. There are fish and then the water from the fourth coastal in Java Island, and we bring that to our laboratory. In the laboratory, for the fish, we will dissect and collect the organ that we need. And for the water, we will put it in Erlenmeyer and then we bring that to the oven. We will use for about 60 under 70 degrees until it dries. And then the third, we will give 20 millimeters of hydrogen peroxide to dissolve the organic compounds. Next, we put it in the oven in 50 under 60 degrees for about 24 hours and strain it with a vacuum pump. Then we will put the Wokman in the oven at 45 under 60 degrees for 30 minutes. And then the next step is we will see the Wokman under the microscope and then check the material component using the FTIR. And then the next is how I can get a conclusion for my research in here. The first, for the abundance, my data will be processed by the Mashura et al. In 2015 with the pattern is for the water, the total microplastic that we found per volume, and then for the organ, the total of microplastic per gram of the organ. And the second, we will use the statistics test using SPSS 24 with the normality test, ANOVA, and chi-square. So we will know about the correlation between the microplastic in the water and in the organ of the fish. And also for the FTR, we will use the base data of FTR to make us sure that the sample is the microplastic. And here is my reference for make this presentation today. And I'm sorry if there's a lot of mistake and bye bye.